everybody. So thank you very much for coming. It's a Friday morning. There are a lot of things that you can be doing with your time. Today you're about to hear about bees. And I'm hoping that this will be about honeybees in a way that you haven't heard about them before, because bees have become very popular in a lot of ways in cultural consciousness lately. But there's some things that have been overlooked for some time that I think we really need to get aware of. Our honeybees have been in a state of health decline for some time now, and it's actually a result of a spread of infectious disease from certain places in the world around to the rest of the world. And in a lot of ways, it's been akin to the pandemic that we all went through and are still going through. And so now that I've been talking about this as our pollinator pandemic, it's been getting people to listen a bit more. There are magical bees wearing masks and everything, and I think it helps. Without further delay, I'd like to jump into this presentation in detail to some of the work that we've been conducting so that we can stop the current pollinator pandemic and potentially stop the future ones that are on the way. So, the global health decline that I mentioned before, it's become very concerning. We reached a point where we thought that things wouldn't get worse, and then they got worse. So, currently in the Western world, we lose between about 34 and 52% of our honeybee colonies every single year. That is staggering. Those numbers are absolutely unsustainable. And so we've been trying to figure out exactly what's causing these issues and what we can do about it. Now, we know that there isn't just one thing going on. There seems to be competing, interconnected factors all coming together here to create a problem. But there is just one issue that tends to take up all the air in the room. We know that chemicals can have a pretty dramatic impact on insects. And so for the most part, when people talk about what's going on with our honeybees, they focus on pesticides. And with good reason. Chemicals that are designed to kill insects kill insects. Problem solved. We can all go home now. Thanks for listening. That's all. Bye, everybody. But turns out things can be more complicated than just that. Now, we've realized that this decline has been noted across multiple species. There are 9, depending on who you ask, there are between 9 and 11 species of honeybees. Some researchers are still arguing about this. They can age up over it, so I'm not going to get into that argument. But there are currently confirmed 9 species of honeybees. And to some extent, all of them deal with this whole suite of factors, from pathogens, pesticides, climate change, human conflict, habitat destruction, poor nutrition, and parasites. Now, the largest study ever conducted, the largest conglomerated study that we've ever seen, was conducted by the United States in an attempt to understand why our honeybee population was declining the way that it has been declining. And what they found is that the 3 biggest issues are pathogens, pesticides, and parasites. Now, the biggest of the 3 P's, as we call them, is parasites. The parasites, at the time, weren't fully able to account for why the parasites were such a big deal. But we're learning more and more every day that these parasites cause dramatic issues for the bees. And the parasite are the one element in this system that's connected to everything else in this system. So, the parasites are actually vectors of pathogens. They can vector different viruses that are really problematic for the bees. The parasites also feed on a section of the bee that detoxifies pesticides. So any pesticide that they're exposed to is much more lethal than it would be otherwise if parasites are present in the colony. The parasites also make it more difficult for the bees to adjust to perturbations in the environment. So it's more difficult for them to adjust to climate change and to the fact that a lot of their food doesn't have the nutritional content that it once had. And it makes it more difficult for them to navigate and fly long distances. So as their habitat is destroyed, or as they come in contact with more and more humans and human conflict, it's much more difficult for them to adjust to that and move to other locations. So the parasites have seemed to be this connecting factor to all the other things going wrong with the populations of bees. This mounts up to a huge issue with stress. Our bees, like us, are really stressed out. A huge issue, you know. is that when all of this stress comes together, it weakens their immune system and it weakens their ability to suppress parasites. And so you'll notice that in colonies that are very stressed out, their parasite numbers go through the roof, and it typically leads to the collapse of that colony. It is also really concerning because when colonies are very stressed out and the parasite levels get really high, that can make that bee a reservoir for more issues within the environment, because as they end up with tons and tons of viruses, they still visit flowers. When they visit those flowers and sip the nectar, they deposit those viruses into the nectar, such that other species of bees that don't even have those parasites, solitary bees like carpenter bees, can get those viruses, and oftentimes it is lethal for them. While in a colony, if there are 20,000 bees walking around, if a few of them get viruses, the colony can still survive. With the solitary bee, it's the end of that organism's life. As these parasites and viruses mount, they can allow for the bees themselves to become vectors of these diseases to other regions of the world. Case in point, the pollinator pandemic that we are currently dealing with is a result of this parasitic mite that you are looking at right now. This is Varroa destructor. There are actually 3 mites on this bee right now. One is tucked in between these layers of the bee's abdomen and is feeding on it. This parasitic mite feeds on both the adult bees and while they're in their juvenile stage and developing into adults. And much like COVID-19, this infectious agent jumped from one organism that it was already a part of their evolutionary sequence to a totally different organism that had no evolutionary background for dealing with it. So we believe that COVID-19, because it's most closely related to a bat version of the virus and a pangolin version of the virus, we feel that there at some point was a jump between these organisms and eventually to humans. Similarly, we know that the versions of the parasites that we are currently seeing spread around the world came from Apis cerana, specifically in Thailand and Korea, the ones that are most broadly distributed around the world. And this is really interesting because the same sort of spread that we saw in how an infectious disease like COVID-19 spread around the world, we saw something very similar as this parasite went from country to country to country, causing heightened losses in honeybee colonies. In the countries where this parasite is present, losses are between 18 and 68% each year for their honeybees, which is an unsustainable amount of bee death. Recent breakthroughs have actually helped us understand better why these parasites are such a big deal for the colonies. Now, this creature, just in case you didn't get a good look at it earlier, I want you to realize you are currently staring into the face of evil. There it goes right there. That organism is known as Varroa destructor. And if you're having a hard time seeing that face, let's enlarge it for you. That creature right there, that organism is causing more damage to bees than any other factor that we know of right now. Because it has such an effective means of getting into colonies, remaining in colonies, it's actually able to chemically cloak itself in the smells that the bees utilize to recognize other individuals who are supposed to be in their colony. So it can make itself smell like a bee. Because it's inside of a colony that's totally dark, it doesn't need to look like a bee, but it does need to feel like one. And if you could look even more closely at the hairs all over its body, you would see that they branch in the same way that the hairs do on a bee's body. Now, bees have branching hairs so that they can collect pollen. These creatures don't collect pollen. They do not do that. The only point for them to have all of these branching hairs all over their body is so that they feel like a bee when the bee is looking for something on its body that needs to be removed. So these parasites have done a great job of getting into the colony and exploiting their host inside of it. This invasive species was first found in the United States in 1987. I had nothing to do with that, by the way. I was -2 at the time. But when they arrived, we knew that we had a serious issue because within just 10 years of its arrival, it killed 99.98% of our wild honeybees. So that's kind of scary to be able to accomplish that in 10 years, but this is what happens when you have this jump between species.
Are you freaking that there? Are you freaking that?
Yes, so this In the US, we only have Apis mellifera, so the European honeybee. The wonderful thing about Southeast Asia is that you guys have every other species of honeybee present. We just have one. So if we lose ours, we're doomed. But you guys, you'll be fine. The important part is maintaining the biodiversity at the heightened levels that naturally exist in the environment, because that's what the ecosystem evolved with. Losing even one of the links in that chain can be deeply problematic. So this parasite was originally discovered in Indonesia. It moved to China in the 1950s. Around 1959 it was discovered, and then started spreading around the world, eventually ending up in our neck of the woods. But when it was just in Southeast Asia, it was feeding on bees that have spent tons of evolutionary time with it. And so they were able to control this parasite. It's when the parasite's levels grow so much that they start sniffing around for other hosts that things become problematic. And we're starting to see that in other species of parasites now, as they're crossing over from Apis florea bees to Apis mellifera. Some are crossing over from the giant honeybees down to Apis cerana, and it's become this whole exchange of parasites and viruses. Now, this is the part where I really enjoyed talking about just how interesting the scientific process can be, because there's a mystery going on in the video that you're looking at, right? This parasitic mite that you see attached to the body of a honeybee is doing something that for the last half a century we haven't been able to pin down. We were under the impression that these mites were feeding on the bee's blood, based on a paper that was written in the former Soviet Union in the mid to late 1960s. And this paper was written in Russian. This paper was very difficult for people to find and was only published in a very obscure journal. But one researcher in the US cited this paper and said that it proves that that the mites are feeding on the bee's blood. That was actually a mistranslation. And we've been walking around talking about this parasite feeding on the bee's blood for the last half a century. And the concerning part there is that we've been very confused as to how the 9 different diseases that result from this parasite feeding on the bees could result from them removing a small amount of the bee's blood, when every single day the bees make more blood. So what exactly is going on with this parasite? Well, when I was a graduate student a long time ago, I decided that this was a subject I wanted to tackle and better understand, because it seemed like, because I had looked up this ancient paper, I was the only person who had actually read this in recent history and could tell that that paper never proved what it said it proved. So I conducted some research on the subject, first just observationally. Where do the parasites feed on the bee? You can actually learn a lot about an organism from where it chooses to feed on its host. If they choose to feed anywhere on their host's body, well, then they're probably feeding on a tissue that's ubiquitously distributed. Something like skin or blood, which is pretty much everywhere on an organism's body. But if you find a creature that can only feed in one spot, then they may be localized to that area because they're feeding on a tissue that's specific to that location. So what do we find? This heat map shows that the mice only feed in one spot, on the underside of the bee's abdomen. So my question became, what exactly, what's hanging out in that region of the bee's body? If you'll notice here, the mice insert themselves between the plates that make up the honeybee's body, they stick their mouth parts through, and they start the feeding process. And so if we look on the other side of that tissue, what do we find? Well, this is an electron micrograph image. It took me like ages to get, and so I'm going to go back because there weren't enough oohs and ahs when I unveiled it. Ready? Set? Thank you. Very much appreciated. No, seriously, some of you already know in this room when you're a graduate student and you work on this stuff for like hours and hours and hours every day, and then you finally get it. It's really exciting. This took 66 tries, by the way, but we put a honeybee that had a mite on its body in liquid nitrogen. So they're frozen down to negative 172 degrees Celsius. And then we hit them with a really, really thin knife called a feather blade that causes them to fracture in half. And then you can see the internal contents of both organisms. And what you'll see here is this tissue, called the fat body, is surrounding the mites in the region of the body where they choose to feed. This membrane right here is called the intersegmental membrane. It's very, very, very thin, and that's where the mites push their mouth parts through, and the only thing that their mouth parts will be in contact with on the other side is this fat body tissue, which looks pretty similar to the stuff that you see in their digestive system. So in order to actually determine what these parasites are feeding on and why they have such a dramatic impact on the bees themselves, I did something that I think every little kid dreams of. Like any little kid who ever wanted to be a scientist has seen the little clip art pictures of like an old guy holding up two glowing tubes. I got to be that guy, but just like way more brown. And assume because I got to feed glowing liquids to the honeybees to stain the tissues that are in that region of their body. And then the idea was to look at the mites, to actually dissect the mites open and see what color the mites are glowing. So we stained the bees' blood yellow as a control to see are the mites actually feeding on the bees' blood as we've been saying for the last 60 plus years. Or are the bees feeding on the fat body tissue? This sample of fat body tissue that has been stained with Nile red. It is a tissue-specific biostain. It attaches to fatty tissue and only glows when attached to these non-polar substances. And so, expectations. If the mite is glowing yellow, it's been feeding on the bee's blood. The mites are glowing red, they've been feeding on the bee's liver. This tissue, called the fat body, is actually the liver of the bees. For most people at this point, like, bees have livers? What? But yeah, they got it. The liver is really important for detoxifying things. The liver creates all the proteins in the organism's body, the same way that it does for us, not all of them, most of them. So, check that out. This experiment we conducted 68 times, and every time these mites showed very clearly that they have been feeding on the liver of the bees with this bright red glow associated with the biostains that we're feeding them. And so we're learning just now, after decades of dealing with this parasite, we are just beginning to learn how it actually impacts its host. And that is kind of scary. Because if we really want to understand how to keep our bees healthy, we need to understand the things that are making them unhealthy. And the most fundamental way that a parasite interacts with its host, the baseline thing that you have to understand if you're going to understand a parasite, is what it's eating when it attaches to its host. Now, I knew since I was still a graduate student, new to the scene, I'm just jumping in here, I really got to dot all of my I's and cross all of my T's, and so I wanted to conduct one more experiment to show for sure, because I was sure that when I submitted this for review, one of the reviewers would say, hey, you've shown that the mites consume some of this liver stuff, but you don't know that they need it. Maybe it's just an accident that they're getting the liver in their mouths. Maybe they actually need the bee's blood. And so I conducted a study where I actually fed the mites. I created this cute little, do you guys notice one of these things is not like the other? Yeah, okay, all right, I was hoping you would be fooled. But the mites were actually fooled, which was really exciting. I actually created a decoy bee from size 5 gelatin capsules that I found in a pharmacy and figured out that they're pretty similar in size to the bees themselves as they're developing. And then I took a very, very, very thin layer of something called paraffin and stretched it over this to create a membrane so that the mites could feed through it, and the mites wouldn't feed for months. It was very frustrating. I got really cranky. I got to a point where I was talking to the mites, like pleading with them in the lab. Come on, guys, just eat. just a little bit, just sip it. And then I thought to myself, oh, Sammy, you are being such a doofus right now. You have thought to get something that is similar in size and shape to a bee as if the mites have eyes. They can't see. They're totally blind. It doesn't matter what this thing looks like. It matters what it smells and tastes like. So then I started taking the parafilm and rubbing it on the bee's body so that it smells and tastes like a bee. And bingo! the mites started feeding. I was able to start these experiments where we confine them with these decoy larvae and allow them to feed and lay eggs. And then I could inject these decoy larvae with fat body, like the liver tissue, or hemolymph, or a combination of the two, and see which of these does the mite actually need to grow and develop and produce offspring. It turned out, in order for them to actually make these gigantic eggs that they make every day, this egg is huge by comparison to this female body, about 38% of her body volume. So that would be like a woman who weighs 100 pounds, having a baby that weighs 38 pounds every single day, which is terrifying. But these parasites are able to do so, but only when they are fed the liver of the bees. If they are fed the bees' blood, they don't lay any eggs. And so we were able to publish this work, but what was really helpful for us was to actually understand what is happening to our bees. This tissue that you're looking at is the fat body tissue. This is a dissection of a honeybee. Don't worry, she died of natural causes. We were able to cut open and look at the liver, and you see this huge tissue. All of this is the bee's liver. And anytime you see a tissue that takes up that much space in an organism's body, you know it's doing really important stuff. So it's handling the growth and metamorphosis of the bees. When the bees need to transition from a larva into a pupa into an adult, all of that is regulated by the hormones and proteins produced by the bee's liver. In addition to that, storage of the different elements of their energy and nutrient mobilization is all very important and is managed through this. So when these mites feed on the bees and they disrupt this tissue, it makes it very difficult for them to manage their own metabolic activity. And so they'll have the energy to fly to a set of flowers, but then the foragers don't have the energy to come back home. And you'll end up with colonies lacking in much of the workforce that they need in order to survive. Even things like water loss is a result of the... Really? You guys are scared of me, are you? No? So, I don't do trick questions in my presentations. Just letting you know. Actually, this is going to be, so my dad's a pastor. This is going to be a lot like Sunday school, if you've ever been, where it's like, oh yeah, what's the right answer to this? Like, the right answer in Sunday school is always Jesus. Like, in this situation, the right answer is fat body. So, the fat body, the bee's liver, is the tissue that manages their immune function. And the tissue that manages temperature regulation, that is... It is the fat body. And we'll accept that answer too. The fat body tissue is incredibly important because it also manages the immune system, such that when these mites are fed on, or when these bees are fed on by these mites, their immune system plummets in its ability to handle and protect these creatures from immune compromise. And temperature regulation, protein and fat synthesis, all of these things are regulated by this one tissue. And so it's really problematic that the primary sets of parasites that we find inside of honeybee colonies distributed around the world are attacking this tissue specifically. And we just figured this out. So why is that important? Well, that's important because Varroa destructor is not the only parasite that our bees are dealing with. While Varroa destructor focuses on the European honeybee, there are a lot of other parasites. Let me take you on a little tour through the world of parasites of honeybees. We've got the greater wax moth, the lesser wax moth, fungus beetles, Alphatobius litigatus, Alphatobius diaphoratus, the history of beetles, small hive beetles, bee lice, Varroa, also Megavarroa, the giant bee lice, the Varroa mites, 4 species, the Euvarroa mites, 2 species that we know of so far, the Tropolalaps mites, 4 species, pollen mites, Pseudasia mites, leptis mites, predatory mites. Quite a lot going on here. Now, the concerning feature here to me is that there are 2 or fewer papers written about the majority of these organisms. And that is disturbing. Because the greatest impact on our honeybee colonies right now that we really have to make sure that we are understanding is that there are symbiotic relationships between organisms. There are the good symbioses or mutualisms where the creature is helping the other creature, and some of these organisms may actually be symbiotic. Maybe. But then there is also parasitism, where the host is heavily impacted, and parasitism is a system that works really well if a creature learns how to adapt to its host. It can fully exploit them and cause all kinds of problems. So we have 9 species of honeybees and all of these other creatures trying to get into their colonies. And it can seem crazy to even think about that being the case until you recognize a honeybee colony is just the best place to live. I mean, it really, really is. If I were small enough, I would find my way inside of a honeybee colony right now. So I recently moved to take the professorship that I currently occupy at the University of Colorado Boulder. And so I spent a lot of time recently looking for apartments and houses and things. And you quickly learn there's some stinkers out there. Not everything is a great place to live. Well, honeybee nests are the perfect home for a number of reasons. One, food availability year-round is an amazing thing. In a lot of regions of the world, there's a level of food scarcity that comes along with the winter or the rainy season. The bees have stockpiles of food that constantly allow them to be active year-round. And if you can be an organism that makes its way into the honeybee colony, you have access to that year-round food. In addition, the colonies are constantly environmentally regulating themselves, such that if you don't like the temperature outside, go to a bee colony. It will always be a stable temperature and a stable humidity. And for a lot of parasites, that is the perfect system to live in. In addition to all of that, you have a round-the-clock security detail. If you can find your way into a honeybee colony and remain there, these creatures will use lethal force, typically lethal against themselves, to make sure that they protect their colony and they protect everything inside of that colony as a result of it. So they end up protecting their parasites. People ask me every time I give a presentation, why don't you just find a biological control method of getting rid of these parasites? Find whatever controls them in the wild and then just release it into the colonies and that'll fix the problem. Because everything has something that controls it in the wild, right? Well, not the creatures that live inside of honeybee colonies. Because the honeybees are so good at keeping everything out that they even keep out the organisms that would naturally attempt to eat creatures like varroa mites and leptis mites and mesophilolaps mites and trophilolaps mites. And so that allows the parasites to run rampant and their populations to grow absolutely huge. So the organisms that get into these colonies are called melitophiles. That's from the Greek meli, which means honey, and phile, lover of. So organisms that have created this remarkable symbiotic relationship with honeybees are referred to as melitophiles, and we know very little about them. So that's where I come, especially since I've won the trust of many researchers with the work that I conducted in actually elucidating how the mites are impacting the honeybees. I pitched this big idea that I've had. I want to actually find all of these melitophiles. So I want to find every species of honeybee that exists, all 9 species, and I want to document the symbiotic relationships going on inside of their colonies. By collecting these organisms and sequencing their genome, we can better understand them. And it's a lesson that I think we learned from the pandemic, because I was pitching this idea before COVID-19 showed up. But it wasn't until after COVID-19 happened, and I started reminding people, hey, just consider for a moment, what if there was some amazing compendium that already had the genetic code of COVID-19 in it, and already had all the symptoms, and all the ways that we could treat it already written down? Wouldn't that have been wonderful at the end of 2019 when this issue started spreading around the world? Well, similarly, when we notice these parasites moving around the world, impacting different species of honeybees, if we already know how to manage them and what to do about them, we can much better protect our populations of honeybees. So, I have now been funded for this project through the United States Department of Agriculture and the National Geographic Society, and I'm going all around the world looking at the different species of parasites that impact our honeybees. And so this is from the work that we're conducting in Thailand. National Geographic was so kind to send a photographer with us that we were able to work with and get some really amazing images of the different parasites that we're seeing there. And this is not Varroa destructor. This is actually Varroa jacobsoni, the parasite that goes after Apis cerana and causes substantial issues with them when they become very stressed. We're going to take all of the information that I compiled in this work, that we compiled in this work, me and my lab and collaborators, and we're going to create an open access compendium so that everybody in every country, without having to pay anyone for anything, can access this information and use it to make sure that their honeybees are as healthy as possible. Because sharing is caring, and I think it's ridiculous that we charge people to access the truth, but lies are available for free on all of the social media apps. As a matter of fact, there is an algorithm that will funnel the lies into your face, and we make people pay ridiculous amounts of money to read a single scientific paper once. So we're not going in that direction. So I've been traveling through Southeast Asia, documenting these different parasites, and it has been really exciting. I'm trying to dodge all the mosquitoes if at all possible, because there's some scary stuff like dengue out there. But as we go through and document all these different organisms, we're also documenting the resistance that we see in different honeybee species, and understanding why certain honeybees are resistant to certain kinds of parasites can help us understand how we can protect the bees that are not resistant to those parasites. What exactly are the resistant bees doing? The remaining locations that are still on this list, Japan, Korea, the Philippines, Borneo, Sumatra, Sulawesi, Nepal, Taiwan, Sri Lanka, and Mongolia. We've got, this is a 4-year study, we've got 3 more years to go, so plan to hit up all of these in the next 3 years. I'm going to be a little bit tired. It'll be okay. But this will allow us to see all 9 species of honeybees and all the different, have representation of all the different parasites that we have been trying to better understand here. When we actually get these samples approved, the primary focus that we do in these studies, we collect some adult bees, but because the parasites tend to go after the totally defenseless bees in the colony, they want to find the babies. They want to find the bees that have no eyes, no wings, no legs, no mouth parts that they can use to attack. And then they go into the cell and they feed on them and reproduce. So we've been spending a lot of time collecting brood samples. You can see me and my postdoc Madison right there with our headlamps and our specs looking at these tiny little parasites inside of the brood frame. And in this process, we open the brood cells, and then we're able to extract the developing larva and the parasites that it actually has. We take the parasites back for genetic sequencing, and this whole process, we do our best not to unnecessarily damage any of the brood in the colonies, because for many of these, we can actually put the brood back into the colony after we've removed the parasites from it, definitely with Apis mellifera. If the bees don't want those babies anymore, they actually will eat them and turn them back into protein and make more babies with it. So it's weird and kind of creepy, but circle of life. So we put these parasites into these tubes and take some of the bees as well back for full genome sequencing, because the ones that we find that are resistant to these parasites, we want to know what segments of their genetics allow them to achieve this level of resistance. With new genetic sequencing availability and the power of things like CRISPR, it may even be possible for us to move over some of those resistance genes to other populations of bees, so that we have bees that are much better able to manage their own microbes. And it allows us to have a much better understanding of what's going on when parasites like this start moving around the world. This one is the newest parasite to leave Southeast Asia and spread around the world. So Varroa destructor left Southeast Asia, spread all around the world in about 50 years, and has now reached every country where honeybees are kept, destroying bees as they go. The one that's currently, the one that is mounting, the one that's about to happen, is from this parasite, Trochilelaps racetosii, or the trophy mite. Very few papers have been written about it, but the ones that have been are doozies. In Pakistan, in the 1980s, they were rearing Apis cerana and Apis mellifera bees when these parasites showed up. And they killed all of the honeybees in Pakistan. So all of the Apis mellifera honeybees, they lost every single colony. They lost several colonies of Apis cerana, but thankfully the Apis cerana were able to make it through. But the really concerning thing here is that they lost an entire species as a result of this one parasite and had to send to other countries to replace their population. So we know that because Southeast Asia is a highly diverse region of the world, has the highest biodiversity of honeybees anywhere on the planet. This is also the region of the world where new parasites and new diseases are most likely to emerge. Nearly every parasitic disease that is spread around the world that is impacting the honeybee species has come from this area of the world. I can only think of two exceptions. And so this is the area where I'm focusing a large amount of my research. And anytime we encounter an organism that has the capacity to eliminate an entire population of honeybees, we focus a lot of attention on that organism. So when focusing on the trophy mites, we wanted to understand their life cycle because it hadn't yet been documented. And so what you see up here at the top, this is the very first video ever collected of one of these trophy mites inside of a sealed brood cell. Normally a sealed brood cell is pitch black. You can't see anything. And you might be thinking, how'd you get a camera inside? Technically I didn't. I actually replaced 2 of the walls of the cell with a really high optical clarity glass that I pushed in there, and then focused the camera through that so that we can see everything that's going on in these cells. And then the second segment of the project, the intention is to figure out how we can manage these parasites. So we want to cite the mite and we want to fight the mite. Well, this process actually allowed us to understand a lot about the trophy mites and the varroa mites, the impact that they have on bees, what they're actually doing here. And I just gotta say, I'm only showing this because I'm just super proud of it. Like, this is a really cool thing to me as someone who's been studying bugs since he was 7, to actually be able to see something like this that no one else has had the ability to see. Just made me feel like I've contributed something that is unique. We're seeing varroa destructive or feeding, and all of that white squishy substance you see there is fat body tissue. It's pressing its mouth through the skin of the bee, the exoskeleton, and feeding on that tissue. And we know a lot from all the years that we've dealt with this parasite, that there are 9 different issues that they cause in the bee's body, but eventually, the biggest one of it is that it leads to the bee's death. But something that's also really important to consider is that if we're going to understand a parasite like the trophy mite, we need to be able to conduct this kind of work on this organism and understand all the things that this creature is doing to these bees as they are developing. So this is the first video ever captured of these creatures inside of single brood cells. And we're going to answer the question, what does the trophy mite's feeding do to the bees? How does it impair or impede development? What exactly leads to the death of the bees? Because all we really know at this point is that it transmits viruses that lead to developmental abnormalities and eventually the death of the bees. We also tested ways of controlling those parasites. We actually used solar panels to hook them up to the colonies and actually heat the colonies so that we could kill the parasitic mites inside the colony, because the bees have the ability to thermoregulate while the mites do not. And so while they can change their own internal temperature, the mites don't have that capacity. It was actually a really exciting project and great to be able to conduct this kind of work. And if we encounter any other parasites that are particularly concerning, we'll continue to do this sort of work to better understand how we can sustainably manage them. So all of that to say, I've got just a couple more slides. I want to talk to you about the work that I specifically conducted in Singapore about these organisms. Now, this work was literally conducted this week, so it has not all been analyzed. There is still a lot that we are processing and crunching the numbers on, but there are a few preliminary things that we can talk about here. So the 3 species of honeybees that we really noticed are heavily impacted by parasites and heavily impacted by stress seem to be Apis florea, Apis indreniformis, and Apis mellifera. Now, you guys don't have Apis mellifera here, so you can ignore that. That is the European. But these 2 are the dwarf honeybees that I've seen in a few places here. These honeybees are heavily impacted by stress factors, and it shows up pretty substantially in the amount of parasites, and we expect that when we get to take these back for sequencing, it'll also show up in the amount of viruses that we are likely to see in them. Apis dorsata and Apis cerana actually seem to be pretty resilient to perturbations in their environment. It takes a substantial amount more stress for them to actually show that in their parasite and virus loads. And we know that from our work that's been conducted in Thailand thus far. Now, the samples that we've collected thus far suggest that Apis cerana and Apis florea bear higher parasite loads in Singapore than any other place that we have conducted our research in Southeast Asia thus far. And that was actually kind of surprising to me when I got here, but then starting to realize there were that many colonies here. So the ones that we were actually able to access were from a pretty biased sample. Unfortunately, the ones that we were able to access were probably pretty stressed out. They likely couldn't nest in all the places where they would like to nest. Nests in like the national parks, nests that are very close to human habitation, are likely to be removed 24 to 48 hours after they have been found. But nests, I don't know, off on an island with an oil refinery, let's say, those can probably stick around a little bit longer. And while existing in that space, they're exposed to chemicals. They are, unfortunately, also just dealing with a lot of stress and not being an optimal environment. And so one of the colonies that we looked at had the highest parasite load that I've ever seen in any set of Asian bees that I've looked at. It had a nearly 67% infestation rate, which is unheard of. This was primarily in drone brood, so thankfully the worker brood had substantially less, but still much higher than what is the typical average. And what is the typical average is between 1 and 3%. So that's not great. That organism is existing as a reservoir in that space, which is really concerning. And so I just do want to highlight one more time though, these samples are pretty biased because we don't have a lot of access to random wild colonies just distributed in the environment. But these heavily stressed colonies with their high parasite loads could potentially be reservoirs, not just to other honeybee species, but as reservoirs of viruses. When they land on a flower and unfortunately release viruses into that nectar, that can impact other species of honeybees and pollinators, which can be very difficult for them as well. And so, thank you all for listening to my talk today. I'd also like to thank everyone who contributed to this project. Our funding through National Geographic, the USDA, Honeyflow, and Manuka Doctor, as well as Project APIS-M. And if there are any questions, I would be delighted to answer them. Woo! So, while you guys are gearing up for any questions that you might have, actually, we've got like 15 minutes left. I'd like to set up with a frame of honeybees that I collected while here in Singapore and show you guys what this process is like. So don't worry, I can walk and chew gum at the same time. Feel free to start asking some questions, but I want to show you how this process actually works, evaluating disease spread in honeybees. It's a big opportunity to actually look at the most vulnerable organisms in the colony. The most vulnerable bees are the ones that are right under these capped cells. The bees literally place a cover over them to protect them from all the ne'er-do-wells out there, all the creatures that could wish them ill. And the parasites, some of them, have figured out how to get around that protective capping. So right before the bees place that capping, what triggers them to understand that it's time to cap that cell is a set of volatiles, smells that come from the developing bee's body. The little baby curled up inside the cell releases chemicals that let the bees know to cap the cell. The mites have figured out that same smell, and they scurry into the cell really quick while the bees are putting the cap on top of it. And then after the bees have sealed them inside, they're perfectly protected from everything. We can't get most pesticides into those cells because the wax is hydrophobic. So most stuff is just going to beat up on the surface, and nothing can really get in there with them. They're sealed in there for about 2 weeks. So, I, being the intrepid researcher that I am, put on my super cool spectacles. Let's see. You know, I ask myself all the time, how am I still single? I'll never understand it. never understand it. And I want you guys to be able to see what I see. So let me see if I can plug this in for you. Got some fancy new technology when I became a professor. There are perks to it. Yeah. All right. So, when you're conducting this kind of work, can I borrow some? Thank you very much. I'm going to have to move it this way. Thank you very much. I really appreciate it. Okay. So when you are working with these cells, okay, so when you're working with these cells, the drone cells are very difficult to uncap because the bees put such a huge layer of wax over them. But when you do, actually I think this light is doing more harm than it could. So I'm going to shut it off for that. Okay. So, what we have here is a developing drone larva. You can see that it currently has no parasites on its body, which is great. It's at a stage of development where it still has no legs, no wings, and we refer to it as a prepupa. Yeah. So inside of a cell, if you see a prepupa like this organism is healthy, it's living its best life. So this one is healthy and navigating its development pretty healthily. But then there are others that unfortunately do have parasites. And I wonder if any of them will actually show themselves in this process. Now this one is drone pupa. So this is what a healthy pupa looks like. They have purple eyes at this stage of development. You can see that wings are even beginning to develop on the side. Still not quite used to using this thing yet. It's pretty cool though. And the bees that are walking around on the colony, these are referred to as nurse bees. They don't really have the capacity to do much but walk around on their first day of development. They can't fly or sting. So I promise you, you are in no danger. I can even show you that quite clearly by letting this young lady hang out with me. Look at how they do. Little sweetheart. Look at her. Oh, this is why I'm single. Okay. Figured it out. Figured it out. So do you guys have any questions? I'm going to just keep opening cells and see what we find. So if my presentation was so good that there are no questions. Question. What's up?
So thanks for that amazing presentation. I think really mind-blowing stuff. Great work. My question is about Mellifera in the US.
Uh-huh.
Do you consider consider them to be a native species? I thought they were from Europe, North Africa.
So they are from Europe, North Africa. They are not a native species to the US. We just really like them.
So if non-native species gets affected, wiped out, high mortality rates, beyond this amazing presentation about these bodies and stuff, the impact on ecology, wouldn't it actually, how would you rate a non-native animal getting wiped out in a place that never was, let's say, 1,000 years ago? It didn't belong there, right?
Yeah, they've only been here since the settlers arrived. They came over on the Mayflower, actually.
200 years, yeah.
Yeah.
So actually, what is the very big impact of all the mellifera in the US because they were never there in the first place?
So one of the reasons why you guys probably don't have a strong push for Apis mellifera here in Singapore is because there isn't as much agriculture. But in the United States, agriculture is a huge element of our economy. So honeybees contribute $18.6 billion to the United States economy through pollination, and that's before you even add on the fact that they produce honey and people go wild with that stuff. So it's mostly financial and not ecological. These organisms are not part of the natural ecological, the ecosystem of the Americas. They are actually part of the natural ecology of Europe, and so I'm just shaking everything around now. Sorry, guys. So if we were to lose them ecologically, it wouldn't be that big a deal. Economically, it would definitely upset some people. A big part of it is that we have a very large population in the United States that we feed with a lot of industrial agriculture. And we can't quite do the factory farming levels of food production that we do without Apis mellifera. Now, there is an argument that we shouldn't, that the environment was never meant to sustain that ridiculously large amount of agricultural production, and we should go back to what it was like before they arrived. And I am somebody who could potentially be convinced of that, but I know a lot of individuals in the industry who could not.
Thanks.
Of course.
With the plants like honeybees, and we have the melanomenes here, are you seeing, obviously, are you seeing them in hives, the melamine hives, or are you seeing viruses in the hives that we have to nectar? And also, are there any data or different, within the US, different geographical areas that have mites and might be linked in any way to pollens, helping them to manage population?
Okay, so I did not actually hear the entirety of that question. So I'm going to start with the first part because I think I did hear that, and I'll let you restate the other part of the question. So the first part of your question, I believe, was, am I seeing these same viruses and parasites? Oh, hey, look at you, speaking of parasites. You, young lady, have a varroa feeding on your fat. Does everybody see that little dot right there? The red double?
Can't see that.
Yeah, well, let me see. Is it any brighter over here? Yeah? Do you see the little red dot? Oh, okay. Let me see. Let's see if this helps at all. All right, young lady, you got a spotlight. It's your time to shine. Do you see that? Yeah, you see that, right? That's not a good thing. So she has got a parasitic mite that is feeding on her liver right now, and I'm sure she's not particularly happy about it. She's better on the receptive surface. I'm sure she's not particularly happy about that. That parasitic mite is literally drilling down into the liver just below the surface, and it injects saliva, and their saliva is specifically structured to break down the fatty contents of the liver into a fluid that they then suck out of the bee's body. So it's literally liquefying their liver and kind of turning it into cream of honeybee soup. Which is not great. And I wish that this was not happening to our pollinators, but we are seeing a lot of this in multiple different species.
So from your findings, how long will this be typically live?
Oh my goodness.
Regular lifespan is about a month plus.
Yes, the regular lifespan is about a month. If this parasite doesn't have any viruses that are also going to run rampant in the bee's body, then it may be capable of living for closer to 20 days. So it'll cut the life in half. But if there are also viruses attached to that as well, she's probably only going to live for 4 or 5. So she won't actually get to the point of being a forager. She'll just walk around the colony and do it. I think I've only got like 3 minutes left. This is an hour-long sort of thing, so I'm going to do the lightning round of questions. You asked about the stingless bees, and I love stingless bees. And I have to constantly stop myself from investing a huge amount of my energy in them because my mandate right now is to look at the 9 species of honeybees, and if I get too excited about all the bugs all around me, I will never get through this project. So, for now, unfortunately, the stingless bees have not been able to be a part of this research project, but this project will only be going on for another 3 years. Maybe the next round of it is to check out the stingless bees. We'll see. And then, yeah.
Sorry.
Oh, no, I think he had a second question and I didn't actually hear it. No, okay.
Besides that solar power thing you've done, have you noticed like any effective methods of getting rid of all these parasites besides that?
Thank you for asking. So, for those of you who didn't hear her, she asked, in addition to that solar powered system, I'm going to take this off for a second. In addition to that solar powered system that I discussed, which heats the colony, have we found any other means of getting rid of the mites inside of the colony? And so, of course, we are focusing primarily on cavity nesting bees because it's harder to treat Apis florea or Apis dorsata. They don't really enjoy interacting with me as much. So, but for Apis cerana or Apis mellifera, we have determined that utilization of formic acid can actually penetrate all the way through the comb. So the acid is able to go through the cell cavity into the contents of these brood cells, and it can kill the parasite inside of the brood cell without killing the larva, which is quite remarkable. It was a wonderful thing to find because the trophy mites don't spend much time at all outside of the cells. So the only way to actually manage their population is to send something into the cell to get them. And so we know that formic acid can work. The problem with formic acid in Southeast Asia is that it is very sensitive to humidity and temperature. So if it is above 84 degrees Fahrenheit, what is that Celsius? I don't know. Something Celsius. And if it is above 70% humidity, it can be overly concentrated and can cause problems within the colony, potentially killing some of the brood. And that's what you don't want. So in colder areas of the world, that might be a better option, but I don't think a single day that I've been here it's been less than 84 degrees.
It's 29.
Yeah, thank you. I appreciate that. 29. That's my postdoc Madison, everybody. She's been helping a lot with this project. So, but yes, so we do know of ways of managing it, but we also have to be aware that certain things will work better in certain areas of the world. The heating system is actually probably a better option for this region.
In the penguins now, what do you think of the first vaccine that's come out recently in the US?
Isn't that super awesome? American Falcon. So he asked about the very first vaccine ever generated for an insect, and this is amazing. But we have, the reason why there aren't vaccines for insects is because the vast majority of them don't have an adaptive immune system. They don't have an immune system that can recognize something that's recognized before and then like mount a stronger response as a result of it. But honeybees do. One of the proteins that they produce inside of their bodies just does a bunch of amazing things. It's called vitellogenin or vitellogenin. It slows down their aging process. It's sort of a fountain of youth for them. They stock it up during the winter so that they can live 3 or 4 times longer than they normally do. But it's also a very adaptable protein in that it can fold around other proteins and maintain that conformation shape in a set of, like, basically memory. And they can float around, and if it ever recognizes that thing again, it can attach to it and then recruit cells to attack it, which is basically what our immune system does. So we have now generated a vaccine that utilizes that system to teach the bees to recognize the bacteria that causes American foul brood. American foul brood turns the larvae into a mucus. They develop into about this stage, and then they turn into a blob of green mucus. They look like a booger. And that's the end of their lives. And it can cause an entire colony to rot and die. It's the worst disease that we know of honeybees. It's not super common anymore because in most areas of the world, if you get American foul brood, you're mandated to burn your colonies. And so that's gotten rid of most of it in the world. But this vaccine will prevent people from having to burn colonies in the future. It's very new, so the level of efficacy is yet to be seen, but I'm hoping that it'll be really potent stuff. And I think that I have 2 minutes over, so that's it.
I'm Xavier. I'm a beekeeper in Singapore.
Nice to meet you, Xavier.
I'm not so concerned about Manifera, but I'm concerned about the local bees. So I just want to get it right that local bees do affected by the robot mite and other mites.
Yes.
Just that they are able to better handling them.
Yes.
But when they are under stress, they lost their defense systems. They actually can be infected by the robot mites.
That was actually the perfect summary of my entire talk. You did it in much less time than I did. Well done.
Right. So some of the stress, like when people are removing it with vacuums, using of chemicals, all these are actually causing problems to the local bees. And what I'm seeing is that probably the most being affected is the Primavera. No, sorry, the Adrian colonies. Because we are getting to see much lesser of this colony already. So it may be a problem because of the way that we are handling this.
So I am still pretty new to Singapore, and Singapore is actually, quite honestly, the most unique of any place that I have visited ever. Like, just the ways that everything is managed. There's this very, everything is very, very, very managed. The bees, the trees, the flowers, and so that creates an ecosystem that I can't extrapolate to the way that I could in other places in the world, like Thailand or Vietnam. It's hard for me to make very clear conclusions before I've had time to analyze the data here, but as a preliminary, based on what we have observed so far, it does seem like the bees are pretty stressed. And part of it is because a lot of the bees are probably not able to actually nest in the locations where they would like to. Some of them are being removed from locations, but being rehomed in others. And while that is a great thing, that probably also contributes to stress too, especially in certain ways, like moving them, shaking them around and everything will probably stress them out too. But some of the colonies that I have had the opportunity to look at here, it's clear that they were stressed before they were moved. You can tell this on the basis of the larvae. The larvae are like a snapshot of what's been going on in this colony for the last 24 days. In colonies that have been moved more recently but are still showing signs of stress, it shows that they were stressed out before they were even relocated. And that suggests that there are environmental stressors that are there with them. I don't know what all of those would be, and look forward to better understanding Singapore through future visits. Hopefully I'll get to come back and learn a lot more about the system.
How do you know if the larvae are stressed?
Oh, so that's a great question. How do you know if the larvae are stressed? So there are 3 things, and I probably shouldn't talk about all of them because Zeston's giving me eyebrows right now. So
That's okay, no. Fine, please.
Okay, quickly. So the larvae usually exist inside of these capped cells. I think I turned off the camera. There are some of the cells that are supposed to be, so if the larva is laying down in the cell. There should be a capping over it. That's the stage where they're releasing the smell, the bees come over and cap it. There are some cells that were capped, and the bees have come over and they've removed the capping on those cells. The bees are very stressed out in those contexts. They're running around snatching cappings off of cells because they believe that there's a problem with the health of the brood, and they're trying to manage that system. And you can see that sometimes they go too far, they get overzealous, they cap a bunch of cells. That's how you know a colony is very stressed. In multiple frames that we have looked at of comb, we've seen an overzealous uncapping behavior that's indicative of ongoing stress from the bees. In addition to that, some of the bees that are curled up in the cell will actually flip over early. Bees that are trying to, it's called precocious development. Bees that are trying to turn into adults sooner than they should are also stressed. There's some sort of stressor in the colony and they think if I don't transform into an adult now, I'm going to die. And so they flip over in the cell and they're oriented incorrectly. That is also something that's indicative of stress. And then discoloration. When you remove the larvae from the cells, which I've seen, unfortunately I've seen a fair amount of since I've been here. We collected those larvae so that we can look at what potential viruses or bacteria they have, but that is always indicative of there being some sort of infection when they turn brown like that, and we've seen brown mucousy larvae. We're going to see what exactly is causing it. It wasn't foul brood, I can tell you that, or at least not American foul brood, because American foul brood stinks to high heaven. You smell it before you ever see it. So, yeah. Yes.
From the samples that you have from the samples you think you have a disease problem? I know we have a lot of parasites, there are some companies that parasites,
Yes.
but I think we have some suspect.
Yeah. Her question was, based on what you're seeing in the samples, do you feel that there's a disease problem in the honeybees in Singapore? And unfortunately, I do have to say yes. The parasites themselves are already a disease. They are an infectious agent moving around the colony, liquefying the liver of the bees. You saw one here just walking around on the frame, and I didn't even have to do much to show it to you. It was just there. I didn't plant that, by the way. That was already, it just worked out that way. The concern being, I don't have any of the sequencing stuff here to actually look at what viruses or bacteria might be present, so I don't know if it's one that's already described or something that's totally new. But there is something that's spreading around, and I'm seeing it in more than one species. So I'm seeing it in Serrana and Florida. The Andreniformes that I've had the opportunity to look at thus far have actually been okay. But Serrana and Andreniformes seem like they've been having a fairly tough time. I did not have the opportunity to look at any dorsata hives, and we were also trying to look at the hornets, which can antagonize honeybees as well, and look at their genetics. But we weren't able to find any because people don't like hornets. They sting. So obviously there aren't a lot of them around. But if you guys know of any, just hanging out, I will be back to Singapore again. So, you know, you could just let a brother know. I can grab some of those hornets and do some sequencing on them, and then they can go off into the sunset wherever they want to. But everyone, I would like to thank you very, very much, not just for listening to this presentation, but I'd like to thank the National Park Service as well as the Natural History Museum for partnering with me in this process. The Natural History Museum is the reason why I'm able to actually send these samples